Kia ora koutou, and welcome to the second in our Science Live at Te Papa live streaming events. I'm Ruth, this is Scott, and we're here behind the scenes at the Necropsy Lab in the Museum of New Zealand, Te Papa Tongarewa. And in our Science Live at Te Papa series, which we'll be doing approximately every couple of months, we'll be going behind the scenes in the various science labs we have here at Te Papa, finding out a little bit more about the research that we have going on here. Now, it's a really rare treat for Scott and I to be here in our room. It's quite an unusual location for us, isn't that right?
It is indeed, Ruth. It's a bit different to the office that we're usually used to being in. And the first thing you kind of notice when you walked in here is it's got a very distinct smell, a chemically kind of smell, mixed with the smell of roast coffee, but that's coming from the coffee place next door. The other thing we notice is it's a very clean room, very white. And right in the middle, for those of you who may have been watching our last Science Live event, is our huge sunfish tank. So we'll be walking around and navigating our way around that tank today as well.
So in today's live stream, we'll be finding out about a group of seabirds that was particularly affected in a huge storm off the coast in July 2011. Now, many seabirds were affected by this, but particularly affected were a group called Prions. And you may also know them as whalebirds, and we'll be finding out why they're called that later in the stream. Now, there are about 6 species of prions, and there's some particularly beautiful Te Reo Māori names for them. We have the broad-billed prion, which is called the parara, after its kind of croaking, rattling vocalizations that it makes. We also have the beautifully named Titiwai Nui, which means little seabird in the big ocean. And as I said, these seabirds were washed up on the coast of New Zealand in July 2011, and it was many people's, ours included, first experience with these birds. For the majority of their life, they spend their time out on the Southern Ocean. So although they're one of the most numerous birds in the Southern Hemisphere, many people never see them. And in this huge storm in July 2011, over 300,000 of these birds washed up on the west coast of New Zealand. Some of them even blew into people's doorways and into their gardens. Tragically, many of these birds were dead or dying from exhaustion. And many of you, the viewers, may have taken them to your vets, to universities, to the SPCA. And through these various channels, they ended up right here in Te Papa. And that's what we'll be finding out more about today.
That's right. Over the next hour, we're going to be talking to some of our scientists here at Te Papa to learn a bit more about the birds, about the storm, and some other things as well. And if you have questions that you might want to ask during the live streaming event, you can contact us via Twitter using the hashtag Science Live Te Papa or sciencelive@tepapa.govt.nz. You can email your questions there as well.
Thanks, Scott, and we're really looking forward to hearing your questions, so please do send them in. If we don't have time to get through them all today, we'll probably write a blog about them, or we'll get back to you later on as well. So I'll say goodbye to Scott now. He's off to chat to some other scientists, and I'd like to welcome in Susan Waugh.
Hi Ruth, how are you doing?
Good, thank you. Hi Susan, lovely to see you. So in case you don't know, Susan's the Senior Science Curator here at Te Papa, and handily for this episode, her speciality is seabird foraging, so that's very convenient. So what a lot of people might not know is that we do science every single day here. So would you be able to tell us a bit more about the science that goes on?
Sure, we have 10 science researchers here. Some of them are curators and others of them specialise on different topics that aren't to do with the collection, such as genetics. And our researchers are mainly working on taxonomy topics, so differentiating one species from another and describing those species. And we describe a number of new species to science every year. Over the last 10 years, we have described around 250 new species, including some birds and whales. So it's really quite an important group in terms of covering lots of different sorts of plants and animals. But also we help underpin crucial things like whether species are new in New Zealand, whether they cause a threat, or whether they should be here, and they've always been here in our biota.
So it sounds like there's lots and lots of interesting things that go on, and it would be a really fascinating place to work. But what's its value to maybe New Zealand or to the rest of the world?
So the science that Te Papa contributes, it operates in lots of different ways, as I said, to do with biosecurity or just in terms of describing the uniqueness of New Zealand's biology, but also things about which species of fish, for example, we're fishing, whether some fish found in the north of New Zealand are exactly the same species as those in the south, which helps us make good decisions around using the fisheries.
Okay, so that sounds really interesting then. And what do you do on a daily basis? What does your job involve?
So I have a role that looks at how we do science strategically in Te Papa, connecting our work to what's going on across the sector, working with other institutions like universities and the Crown Research Institutes like NIWA and Landcare Research. But also I help look at the whole of our science program and make sure that we're addressing the really big questions for New Zealand science and that we're on track with delivering our work.
And so in your time at Te Papa, what's the favourite thing that you've discovered or the favourite piece of research you've done?
Well, I've been involved in working on large seabirds, so albatrosses and petrels, for a number of years, and I recently did some studies where we put satellite transmitters on birds and we followed their movements around the ocean, and that's always really exciting when you do that for a first time for a species, because you discover that they're going much further than you could possibly have imagined, and they're using little nooks and crannies in the marine ecosystem that you think, when they leave their nests and they go out to sea, that it's all one big open playing field, but in actual fact they can navigate their way around these big open oceans and find where there is concentrations of food. It's really exciting, and we've recently done some work where we put out a number of different GPS loggers on birds leaving the colony at the same time, and you can see how they come together and use different wind patterns and how those birds move around in relation to one another through time.
So that leads us really nicely onto prions then, because although you specialise in seabird foraging in Albatross, today we'll be talking about the 300,000 prions that washed up here. So could you tell us a little bit more about the value of that research and what we might potentially find out?
Sure, we've got a number of research programs going on with this group of specimens, and it's been lovely for Te Papa to be able to have access to the specimens, so we're really grateful to the zoos and the SPCAs and other groups that have sent them through to us, because we can use these birds that have tragically died to help us understand where the differences are between populations, whether these birds, predominantly from one species, a broadbill prion, whether many of the birds came from only one location or did they come from many of the sites that that species breeds at. And we also start looking at the relationships between species, how different are they from one another. And we can only really do that kind of study when we have specimens to work on. We can't necessarily do it with live specimens. So we try and make really good use of the specimens in terms of different kinds of science that we do. And as our fish scientist said last time we spoke, that it's a bit like having an amazing library, and every time you take one of the items out of the collection and look at it in detail, like reading a book, you discover new things. You get a different take on the same information, and through time, over the 150 years or so that Te Papa has been in existence, we've used these specimens for many different kinds of science. Now we're doing genetic studies with them, whereas maybe 30 or 40 years ago we would never have dreamed of doing that kind of work. We would have been measuring them and comparing their feathers. But now we're starting to understand things about the biochemistry of the feathers that help us unpick the diet, using genetics from their tissue and from their feathers to help us understand how related they are between species and between different island populations and so forth.
Thanks, Susan. That's a really good analogy. I like the idea of specimens as a library book, and that people can come back 10, 50, 100 years in the future and use them for more data. So thanks very much for your time,
Great.
Susan. It's
Okay.
really nice to meet you, but I think now it's time for us to meet some of these storm-wrecked prions. So let's go across the lab to Scott to see who Scott's talking to. Thanks, Scott.
Thank you, Ruth. So, with me now, I've got one of our vertebrate curators, Colin Miskelly, who's an expert in bird ecology. So first of all, welcome along, Colin. Can you tell us a little bit about what you do at Te Papa?
Yeah, hi Scott. Welcome everyone here to Te Papa's Necropsy Lab. I'm one of the curators of vertebrates here, so my own background is mainly in bird and I suppose reptile ecology, with a strong conservation background. I previously worked for Department of Conservation for many years. And I've actually worked with prions in conservation projects, particularly the translocation of fairy prions from Stevens Island, Tongarewa, to Mana Island, with the Friends of Mana Island who sponsored that work. So we moved 240 chicks and had volunteers hand feeding them, and then they went out to sea, and now some of them are coming back and breeding as adults. So they're a group that I'm very familiar with in the field, at sea and on breeding islands. So it is sad to see these corpses coming in, but it means that we can put that in the context of what these birds do in terms of the rest of their life cycles and try and make sense of this tragedy.
Okay, so it definitely was a tragedy. We've heard quite a lot about the storm, or a little bit about the storm already. We know it's happened in 2011. Can you tell us a bit more about this storm and why it had such an effect on these prions?
Yeah, sure, Scott. I might just grab this map over here. Okay, what happened in 2011, July 2011, is we had a very large storm, and it affected the Tasman Sea for about a 10-day period. And what happened is we had two different weather systems that sandwiched a deep depression south of New Zealand. And so we had very strong southerly winds coming up into the southern Tasman Sea. So this group of birds we think were feeding down to the southwest of New Zealand, down near Macquarie Island. And as the winds blew these birds up into the Tasman, they then rotated around this deep cyclone and pushed these birds mainly onto the west coast of the North Island. So some of the birds were found as far south as Haast and Okarito down on the west coast, and some blew across the South Island, but by far the majority of the birds were found along the west coast of the North Island. But even there, the winds were so strong that some of these birds were being blown inland. Some of them turned up in the Wairarapa, in the Bay of Plenty, and way up high in the Tauruas. So that's actually made it a real challenge to count the number of birds affected, because you can't just walk along beaches counting corpses the way that we usually would for a seabird wreck event like this. We've had to extrapolate a bit for all the other birds that ended up inland.
Okay, so clearly you can't walk along the length of New Zealand and collect birds as you go. So how did we get all these species, all these birds coming to Te Papa?
Well, there's probably two main sources. There's a group called the Ornithological Society of New Zealand, or it's now Birds New Zealand, who have for a long time walked New Zealand beaches, particularly after storms, looking for birds that have washed up dead or dying. And so most accurate figures for the number of birds affected have come from members of the Ornithological Society. And in some cases, the number of birds coming up was in thousands per kilometre. At the peak areas, that means roughly one bird every 1 or 2 metres. So it was just phenomenal numbers of birds that were affected at some sections of New Zealand coast. But overall, the estimates are something up to 300,000 birds that were affected. Then the other main source of specimens that came in, particularly the ones that were recovered live, was via SPCA and Department of Conservation, veterinary clinics, and particularly Massey University, where they've got a wildlife health unit, and Wellington Zoo. So we know that Wellington Zoo alone cared for over 600 prions at the peak of this event. So it put a lot of strain on their resources too.
Okay, so 300,000 birds affected at an estimate. Obviously, we haven't got space for 300,000 birds at Te Papa. How many birds do we have here with us today?
Right, well, we've got just under 900 specimens that were recovered from the wreck, and we've got some of them over here. So how about we go have a look?
Let's go and do that. So we're standing in front of a very large freezer here. I assume this is where you keep the birds here?
Yeah, that's right, Scott. And I'm just putting some gloves on here because these birds, a lot of them have been dead for quite a while, and so they do carry a variety of bacteria and other nasties. So we're always fairly cautious to put gloves on, not only with the specimens when they come in freshly dead, but also with the specimens that have been prepared as study skins, because they've got various toxins on them. Plus, we don't want the oil from our hands to damage the specimens long term as well. So just open the freezer and you can have a look at just how many of these birds we've got here. So all these buckets are full of dead prions. So we'll just pull one out for today.
So we'll get one out. Looks like there's probably, 15 buckets in there?
Yeah, as I said, it's just under 900 specimens that we've kept out of the many thousands that were offered.
Let's have a look at how many we can have fit in one bucket. Okay, so that's a lot of birds. That's from one bucket, and I'd say there's probably 20 or 30 birds in there, and you saw that we got about 15 other buckets, so yeah, quite a lot of birds.
Yeah, well, one of the first things we did was we had to sort out which species were involved, and as we'll hear shortly, there are 6 different types of prions, and we've got examples of all of them here. But the main species that was affected by this wreck was a species called the broad-billed prion, or parara, which, as its name suggests, it has a very broad bill. And again, we'll talk a bit more about that over the course of the next hour. So we estimate that something like 90% of the birds that died in this one storm event were broad-billed prions.
So there was six species altogether, but the majority of them were broadbills.
That's right, and this is a species that does breed in the New Zealand region, so their main colonies are south of New Zealand, around Stewart Island, and also on the Chatham Islands. And that's one of the things that we're hoping to find is whereabouts were the populations that were most affected by the storm.
Okay, thank you very much, Colin. I think that was a very good introduction to the storm and the number of birds that we have here at Te Papa now as a result of that storm. We're going to go and cross over to another curator now and find out a bit more about the different species of the prion. So thanks again, Colin.
Thanks, Scott.
So with me here now is Alan Tennyson, another curator. So first of all, welcome along, Alan. Can you tell us just a little bit about what your job is at Te Papa?
Yeah, I'm another one of the vertebrate curators like Colin, but I cover fossils, so bird fossils mainly, but things of bats and other vertebrates. But I also work on seabirds, so I've actually studied prions quite a bit myself, their biology. I did a master's on the calls of prions out on the Chatham Islands where I spent a summer, and before that I'd already spent a couple of summers down in the subantarctic studying prions, so I know prions quite well and quite interested in them. I think they're cool little birds.
Great, you sound like the perfect person to take us through this next bit. So we've got some prion specimens here. We're going to ask you to kind of take us through the different species and see, tell us how we can perhaps tell the difference between some of the different species.
Yeah, sure. Well, of course, as part of one of the main things we had to do with getting all these birds in the museum was identify them to species. Now, this isn't such an easy job because basically there's 6 species currently recognised and they're all pretty similar. So here's one of the broad-billed prions, and you can see, I don't know if you can see that, but basically it's a blue-grey bird above with a nice black stripe going across the wing with a black tip to the tail. And underneath it's white and has nice little blue feet. Now all prions have that same colour pattern. So the way that we distinguish between the different kinds of prions is mainly based around their beak. Now the beak varies in terms of size, mainly the breadth and the overall size. The most extreme is the broad-billed. Now I don't know if you can see this, but the broad-bill's got an incredibly wide beak, and inside it's got these combs along the base of the beak here. And what the broad-billed prion does is take a big swallow of water. It's got a huge distendable pouch in the lower part of the beak, so it swallows up a big, it grabs a big volume of water, and then it closes its beak and sieves out the little plankton on this comb here, and then uses its tongue, its big fleshy tongue here, to pull the food back into its mouth.
So kind of like a whale would eat its food, is that correct?
Exactly, like a baleen whale, yeah.
And is that where the name whalebird comes from?
That's one of the theories, yeah, that it has the same feeding mechanism. Another is that basically they followed whales because whales created a lot of disturbance in the ocean, and even maybe whalers that killed whales was debris floating around which attracted the prions scavenging food. So, yeah.
All right, interesting. So let's have a look then at some of these different species.
Yeah, so in terms of distinguishing the different species, as I said, it's mainly based on size of the beak. So at the extreme end we've got the broad bill here, which is a very broad bill. And next we've got the Salvin's prion. The Salvin's prion's only from the Indian Ocean. It also has these combs on its beak, but some of these fly over here in the New Zealand winter. Then there's the Antarctic prion, which has a slightly narrower beak again. The Antarctic prion's a very widespread species. It occurs all around the southern oceans, even down onto the continent of Antarctica itself. And then the narrowest of all is called the thin-billed prion, which has a pretty narrow beak. It's also the palest of all the prions. It's got a whiter face, as you can maybe see between these two, and it has a paler tip to its tail. And then we have the smallest two species, which are the fairy prion, which is this one here, which is a very common bird around New Zealand, but it also breeds in other islands around the Southern Oceans. It has a particularly black-tipped tail. And then there's the rarest of all the prions, the fulmar prion, which is very, very similar to the fairy prion, but has a slightly bulkier beak and also has the large black-tipped tail. Now, the reason for these different beak shapes are presumably because the birds are feeding on different things. So the ones with the pointier, smaller beaks are picking up individual prey items mainly, whereas the ones with the bigger, broader beaks are sucking in water and filtering out even smaller prey like copepods and things. So, yep. So they're difficult to tell apart, but with experience, you can hopefully get them right. So that was the first part of the dealing with the wreck, was trying to identify which species had actually been killed.
Okay, so I still look at them, they still look very much the same to me. I guess I haven't quite got the experience that you have. So you've kind of identified them all. Is that that's the kind of extent of your role in the project, or is there more to it?
No, well, I was involved at many levels, but one of the other things that the project's trying to work out is the impacts on the populations of the prions. So now, prions, like all petrels, are very long-lived birds. They can only lay one egg, so only raise one chick a year. They can't relay. They may live into probably their 30s. They tend to pair up for life, although it's a bit like people, they don't always pair up for life. Sometimes they might get sick of their mate and move on to another one. But basically, they form fairly stable relationships. They're long-lived, they're slow breeding. So if we can work out whether it was adults or young birds being killed, this is very important, because the impact of killing adult breeding birds is far more significant than if it's just the juveniles getting killed. In the life of prions, most chicks, once they leave the nest, never make it to breeding age. Most of them die at a young age, or they never get recruited into the breeding population. So it's really important that through dissections, which we'll see shortly, we work out whether we've got breeding age birds or juveniles, and also which sex they are, because also if there's a bias, if there's far more males or females getting killed, then that's going to have a disproportionate impact on the population overall. So, for example, the worst impact would be if it's all one sex of adult breeding age that are killed. That would be far more significant on the population than if it was just chicks of both species getting killed. So that's the kind of detail we're trying to get into and work out the true impacts of this wreck on the population.
Okay, thanks. And visibly looking at them, is it easy to tell if they're male or female, or do we have to kind of examine them more closely?
Well, these birds are very hard to tell apart just by looking at them. The males are a tiny bit larger, but it's not really good enough to just measure them. We basically have to open them up and look at their gonads and to be able to sex them. That's really the only proper way, unless we could do molecular sexing, but it's easier for us at this stage just to cut them open and have a look inside.
Okay, thanks for that, Alan. I think, in fact, that's exactly what we're going to do right now, so thank you again for your time.
No problem. Thanks, Scott.
And we're going to switch over again to Ruth, who's going to be working with another one of our scientists to dissect one of these birds.
Thanks, Scott, and thanks to Alan as well for telling us a little bit more about these species. So I'm here with Lizzie Crotty, who's the student intern from the University of Queensland. And what do you think to what Alan said then? Is it as difficult for you to tell them apart?
Yes, I still struggle to tell them apart just by looking at them, which is why we take a series of measurements in order to see which species they are.
So it turns out it's not like trees where you can just count the rings, and it's not like humans where you can just go and ask them. We really need to do a dissection to find out a bit more about these birds, which species died, what age they are as well. So could you take us through a little bit of that process?
Yeah, sure. I'll just put on some gloves.
Okay, as you can see, Lizzie's wearing her safety gloves and her lab coat, so she'll be the one doing all the work today, definitely not me. Plus, I suspect that she's much better at this than I am. So what's the first thing you do, Lizzie?
First of all, we weigh the bird, and these birds Most of them, unfortunately, died of starvation, so they actually do weigh less than some of the birds that I've read about in papers. That they've taken weights from breeding birds. They're a lot heavier, so yeah, these ones are a bit lighter.
And so would you weigh all the birds that come in? That's a standard procedure for everyone?
Yes, it is. So like I said, there are a lot of measurements we take. I'll just take you through a couple of them. So we also measure the length of the wing. And this is one of the broad-billed prions, so it's got quite a long wing length compared to the other species.
So I'm not sure if you can see this, but Lizzie's measurement sheet is exceedingly large, and I can imagine that it's quite time-consuming to measure all of the bits you need to.
Yes, it is a bit, but now I do know this sheet pretty much off by heart, so I can whiz through it pretty quickly. Another measurement we take is the width of the bill, and these being the broad-billed prions, they're quite a lot wider than the other species.
And what are those two holes you can see right at the top of the beak, just there?
These are the nasal tubes, and they're actually unique to this order of birds. So these tubes are used for excess salt secretion because they drink seawater and they've got to get rid of the excess salt in their body. So they do have a solution that drips out of that. They also have a very good sense of smell that they use for finding their friends and family.
So one single set of nostrils snots out the seawater and smells their friends and family. That's pretty impressive, actually.
Yeah.
So they wouldn't use vocalizations to detect their friends and family, they'd just use odour?
Yeah, it's thought that they do a bit, but that's sort of an area that needs a bit more study, I think.
Oh, okay, so that's another interesting thing we could potentially learn in future about these birds.
Definitely, yeah.
So once you've taken the bill measurements, what's the next step?
The next step would be to find out the age and sex of the bird. Finding out the age is, we need to use a series of clues in order to get the age of the bird. One thing we can do is have a look at the skull, because like in humans it takes a little while to solidify. However, most of these birds are too old for us to really use that. Another thing we look at is some fringing on the feathers. So some of the younger birds have a white fringing. But the easiest way is to do a dissection and just have a look inside. So because they have really thick feathers, I've actually already got one I've taken the feathers off.
It's always handy to have one prepared
Yeah,
earlier.
that's right. Yeah. So as you can see, I've taken off the feathers. They have a really, they have thick feathers and they've got a downy patch there that keeps them warm because they spend their lives on the open ocean. So the first thing I would do here, I'll try and make it so you can see. This is the trickiest part really because I'm using a scalpel and very fragile insides. So just make a little incision.
It looks like it's very delicate work that Lizzie's doing now. It's such a small bird with such, I imagine, small internal organs. It must be very tricky to get the bits you require.
Yes, it does get a bit tricky. It gets a lot easier the more you do it, and I've probably done about close to 500 birds now. So when I open it up, this is the area I usually just work out of in order to sex them, because the gonads are actually under all of, so this is all the digestive tract that we can see here. So the gonads are underneath that. So I'd usually just sort of move this out of the way and have a look, but I'll open this one completely up so everyone can see. So here you can see the breast muscles, or what would be the breast meat on a roast chicken. And as I said before, most of these birds died of starvation, so the keel here is very prominent, whereas in a bird with more weight on it, it would be it wouldn't be as sticking out like that. So I'm just going to open this one right up. Sorry about the bone crunching.
So as Lizzie was saying, many of the birds out there did actually die from starvation and exhaustion. So on a healthy adult bird, it sounds like you would expect a lot more muscle tissue, which would obviously add a lot more weight to them.
Definitely, yep. Yeah.
So what is it exactly you're looking for inside the bird?
Well, first of all, I have a look for the gonads, because that's what we really want to find out, the age and sex. So, yeah, first of all, I do that. And then there are other things we can look at, like removing the digestive tract to look at ingested plastics.
So for quite a small bird, actually quite hard work to cut across it all and make sure you can get everything out of there. And I can imagine it's very difficult to find the gonads, actually, being so small in such a small animal.
Yes, sometimes it is very difficult, in particular in juveniles. And I'll just see what this one is, and then I'll show you a diagram as well. Okay, so here we have an adult female, and we can tell that she's a female because right here we've got an ovary with the ovules on it, and here we've got the oviduct, which is the passage that the egg comes out of. So yes, most of these broad-billed prions are actually adult females, our preliminary results are suggesting. Whereas in our some of the other species, such as the salvins, it's juvenile males that I'm mainly finding, which actually are a lot harder to sex. So I'll just show you one here. So as you can see in this bird, a cartoon diagram of it looks like this. So there's the ovary with the ovules, and this tube here is the oviduct where the egg would come out of. And in the juvenile females, you can see that the oviduct isn't stretched; it's just a little thin line. And then in the males, it's a little bit trickier to tell the age because we're basically just looking at the testes size. So the immature males have much smaller testes. The adults, the testes can really vary in size between whether they're breeding or not. So an adult breeding male is very easy to see because they're much larger, but maybe an adult male that's not breeding can look very similar to just an immature male.
So although your diagram has made it look incredibly clear and easy for our viewers to understand, having had a look inside that bird, actually nowhere like as easy as your diagram suggests. So
yes.
I was just going to say I'm very impressed with your bird detective skills.
Thank you. And also, it's lucky we have such a fresh specimen here in order to see the gonads properly, because some of the specimens I look at aren't in very good shape and they're particularly hard to sex.
So after you've done all this research or all the dissection, what would be the next step?
Next step, I usually I take all the measurements and then I take a photo, and we decide whether the birds are going to be used in the collection or whether they are not going to, because we have so many that we can sort of pick and choose the nicer looking ones to keep in order to study later. Yes.
Thanks very much for that, Lizzie. It was really interesting. I am very impressed with your work. I had a small chat with Lizzie's supervisor who said that Lizzie could dissect, measure, do everything she needed to do for a single bird in half an hour, which sounded very impressive. But nonetheless, Lizzie's done 500 birds, I think you were saying?
Yeah, approximately around that, yeah.
So that's a lot of time spent looking at prions. So thanks very much for your help, and I know everyone here appreciates it a lot. So what Lizzie hasn't said is that sometimes with specimen we'll go on to do another step of research involving the tongue, and Scott's here with somebody now who can explain a little bit more about that. Did you see that, Scott? I'm on my way to being a fully fledged ornithologist.
Indeed you are. Very nice pun too. So with me now is Lara Sheppard, a researcher here at Te Papa. Welcome along, Lara.
Thank you.
Can you tell us, before we ask you any tricky questions, can you tell us a little bit about what you do at Te Papa?
Yep, so I'm the geneticist here at Te Papa, and I work with the various curators, mainly sort of helping them out with a bit of DNA data to help solve their taxonomic problems. So since I've been here, I've worked on plants, invertebrates, fish, and also birds.
Okay, quite a varied role you've got here. So focusing on the prions now, it's obviously what we're here to talk about. What's your research involved around the prions?
So we're interested in looking at two things with the prions. Firstly, for each species that was wrecked, we want to see if we can tell whether we can tell which individual breeding colony birds have come from. So, for example, Colin mentioned the two main populations of broadbills are from around Stewart Island and around the Chatham Islands, and we're interested to see whether we can tell where the wrecked birds came from either of those. We're also interested in building a family tree of prions, so just seeing how the different species are related to each other as well.
Okay, so that's what we're doing. How do we extract DNA from a prion?
So we've decided to focus on the tongue of prions because, firstly, if a lot of these birds are in quite bad condition, and the tongue tends to stay in reasonable condition, so it's still pretty good condition for taking a DNA sample. Secondly, Lizzie mentioned some of the birds we actually want to include into the collection. So if we just kind of cut them open and took the liver, it would make like a big mess on all the feathers. So if we take the tongues, then that's something that's not actually in the final prepared skin anyway. So it doesn't really make a mess and it's easy to do.
Okay, so maybe you can show us exactly how we take a tongue from a prion.
Okay, sure. So if we come over here, we select one of our birds, so broadbill. If we just open up the beak, you can see that huge tongue in there. We basically just take a pair of scissors and chop it off, like so. So it's as easy as that, and that's our nice piece of tissue that we can then put in a tube, and it's all ready to take up to the DNA lab, and we do the processing the DNA from the sample up at Victoria University's lab, genetics lab up there.
Okay, so we've got the sample, we send it away to the lab. Can you tell us a bit about what happens to kind of just take the DNA from the tongue?
Yep, so basically what we do is add a whole lot of chemicals, including some enzymes which will break up the tissue, break the cells open. We get rid of all the proteins, and hopefully we're left with quite clean DNA. Then we work out which bits of the DNA we're actually interested in, make lots of copies of those, and then send them off generally over to South Korea, and then they send the DNA sequence, which is a whole lot of letters, back to us, and then we can analyse that. So the whole process probably takes about a week from taking a tongue sample like that to actually getting a DNA sequence back.
Okay, so it takes about a week. Not really quick, but I mean, it's not a huge wait, so it's not too bad. And you were saying you're looking to find out the different populations, so where they were breeding from. Have we got any results back from that yet?
Yeah, so it looks like for broadbill prions, which we mentioned earlier were the most numerous species in the wreck, it looks like we can't actually tell between the populations from the Chathams and around Stewart Island genetically. So unfortunately we can't sort of tell where most of these have come from. Although people visiting the sites have said that around Stewart Island there seems to be a lot less birds the following year, whereas the Chatham Islands there's a similar number of birds, so it looks like we can probably tell it that way. For fairy prions, it looks like we can tell the different colonies apart, so we should be able to have an idea of where the fairies that were wrecked have actually come from.
Okay, so by the end of it we should have a better understanding of, say, of the genealogy or the family tree of the prions.
Yeah.
Brilliant. Thank you very much, Lara.
Thank you.
So we've kind of learned a bit more now. We've looked at ageing and sex in a bird, found out a bit more that we can learn from DNA. Over here, we're going to pass back to Ruth, who's got another one of our scientists, to find out a bit more about what we can find out from these prions.
Thanks, Scott. So I'm here with Sarah Jameson, who's conducting a really fascinating area of research into these birds, because as we said earlier, over 300,000 of these prions washed up on the west coast of New Zealand during a huge storm in July 2011. And we know where these prions landed, but ideally we'd like to find out a little bit more about where they lived, or at least where they spent some of their life and what they ate. But before we get into that, I've just had a couple of viewer questions. So Sarah, if you wouldn't mind answering a couple of these. So firstly, we've got one from Jonathan Christie, who's in the power room at Plateau School, and he would like to know, are prions related to penguins?
That's a common question. Everybody loves penguins. They are actually related, but actually quite distantly. They're different orders, so that means like an order of, well, all the penguins are all in the same order. More closely related are the albatross. So if you're familiar with any of the albatross that you'll see off the coast, especially down in Kaikoura, they're closer relatives. They're also related to like the titi, or like mutton birds that a lot of people like to eat. So they're more closely related to them than penguins, but they do share a common ancestor, but it's just quite old.
It's quite interesting, though, that these birds are more closely related to albatross, which is so much larger than these tiny prions. And we have another question from, apologies if I do not pronounce this right, as it's your Twitter handle, Tereda, who says, there are a few dead prions at Muriwai last weekend. Do you have any idea of the likely causes of this?
It is quite common for seabirds to wash ashore, so without looking at the specimens themselves, I can't really say for certain. But it is fairly common, but particularly after windy days, and we've had quite a few relatively recently, that it could just be that they were blown ashore and they were just not in very good condition, so it was hard for them to fight back and get back out to the ocean. Other causes, sometimes they do get a little bit of oil on them, and that can cause a bit of a problem. So without really seeing them, it's hard to say.
In fact, as you're on a question roll, we've
Bring it on.
just had another couple of questions come in. One of them is from Amy Dello at Plateau School.
Hi, Amy.
And Amy says, why can't you x-ray the prions instead of cutting them open?
Well, funny story. You can actually x-ray them. It is a little bit more difficult to see some of the organs. But a project that I did for my master's, we were looking at the impacts of embedded shot, so little bits of pellets in ducks. So when people shoot them but don't actually kill them. And so we ended up having a lot of ducks there that we were trying to find the embedded shot, and you can spend a lot of time looking for little bits of metal in a bird. So we went to the airport in Greenland, and we're putting about 150 birds through the x-ray there, and it was quite a sight to see for the new tourists coming in. It's like, this is how we roll in Greenland, you know.
Let's not tell Wellington Airport that in case they get a little bit nervous.
Yeah, they might not be so keen.
So as I mentioned earlier, Sarah's research is into whalebirds or prions, and she wants to find out a little bit more about where they live, or at least where they spend some part of their life, and also what they eat. So can you tell us a little bit more about your work?
Sure. Well, one of the problems that we have, because these birds were storm wrecked, it means that basically they didn't eat for days on end, so their stomach's completely empty. So we can't just open them up and figure out what they were eating. So what we're using instead is stable isotopes. And so these are some of the feathers that Lizzie plucked off the birds earlier. And so basically what we're looking at is the chemical composition of these feathers to determine not only where they were when they grew them, but also what they were eating. And so it's a simple story of you are what you eat.
Okay, so in theory then, does this work for any animal? You could take a bit of sample from me, work out I really love pineapples and chocolate cake.
I'm not sure the pineapple and chocolate cake signature, but yeah, technically. I just spent a month in Canada back home, and so literally you could take a piece of my hair and actually analyze the chemical composition, and you could tell that, you know, while I was in New Zealand, for the past 3 years, that little portion there was actually grown in Canada. So you can do it. You look at different elements, but yeah, it's
That's incredible to think that your hair will grow as a signature of where you spent some time. So how exactly does your research work then? What do you have to do with the feathers to find out that kind of information?
It's a lot of nitty-gritty kind of work. But what we basically do is take each individual feather, and it gets washed just manually, so just with water. And so what we're trying to do is remove any of the dirt and compounds that are on it. And then after that, we go through a series of washes, first with a detergent, and that'll remove some of the more, the stronger grit, the stuff that would need to go to the washing machine. And then after that, we wash it with some solvent, so that'll remove some of the lipids. So basically what we want to do is get the true signature of the feather rather than what's on the feather. And then once we do that, we go to our partners at GNS, and they have Stable Isotope Lab. And what I end up doing is measuring about 500 micrograms of the feather, which is not that much. And then I take that sample, and it gets placed in a tin cup. And so just a little tiny tin cup. So it almost looks like a little piece of aluminum foil. And then I put the sample in there and then fold it up, and that gets fed into a mass spectrometer. And the mass spectrometer basically incinerates that and reads off the chemicals that are released during that process.
Okay, and for those of you that don't know, GNS is a Crown Research Institute here in Wellington, and that's where Sarah goes to take her samples. So what chemicals are you looking for when you get that information back off the machine? You've got all this data. What is it exactly you want to find out?
Well, the general question that we're looking at with these feathers is looking at the carbon and nitrogen signatures. So each element, so like carbon, nitrogen, oxygen, they're all elements. So each one of those has multiple forms. And in nitrogen, say you have two different forms, the N15 and the N14. So N15 is heavier, and the N14 is lighter. So what we're looking for is a ratio of those two isotopes, so two different forms. And as I'll show you here. But as it goes through the ecosystem, it gets past, the actual composition changes a bit. So you have a little crustacean here, and as this fish eats them, he takes in the same nitrogen from that crustacean, but when he passes it, he actually passes out more of the lighter ones. So as it goes through the ecosystem, the heavier isotopes kind of starts accumulating. And so as you go through the whole trophic level, so you're starting down all the way to the phytoplankton, so basically the plants of the ocean, the small plants of the ocean, all the way up from the herbivores to the predators to the top predators, those heavy isotopes start accumulating. So what we're trying to do once we get a reading from this feather is more or less trying to see where it fits in. You know, are the prions the top predators, or are they all the way up here? Or are they actually just more or less eating a couple of the herbivores? Would they be down there?
So from your research so far, where do our 6 species of prions fit into that list?
Well, they'd be relatively low. I mean, they're not, eating things like sharks or anything like that, so they're quite a bit lower. So what they're feeding on primarily are more of some of the smaller invertebrates that we have, so isopods, copepods. But because we have the 6 different species, what we want to try to do is tease apart, by looking at their bills, they do feed on different things. So this is kind of one of the ways to answer the story of, which one's feeding on what. So we're figuring it'll probably be relatively low.
So what kind of stuff do prions feed on then? Is it generally small crustaceans or anything else?
It's primarily small crustaceans. They eat, amphipods, copepods, copepod, barnacles, pretty much anything they can fit in their little gullet.
So what else has your research told you then? Because you mentioned, Lizzie mentioned earlier something about plastics. Prions have been eating plastic. So can you tell me a bit more about that?
Yeah, so one of the other lovely samples that Lizzie collects for us is that she takes the entire digestive system. So basically what that is from when the bird swallows some food, so it takes all the esophagus. And birds don't have a typical stomach like we do. They have a softer stomach called the proventriculus, and then a very muscular thing. That's what grinds the food, and that's called the gizzard. So she takes from the mouth and then all the way down to the bottom of the intestines. And what we're then do is actually open all that up and look for plastic content. And so birds, because they're foraging on the top of the ocean, they tend to feed on basically any floating particles that are up there. So we're trying to see whether or not they're ingesting plastic.
And what's the problem with ingesting plastic? Do they just excrete it, or is it actually quite a serious problem for a bird of this size?
A bit of both. So they probably do pass some of it, but particularly because of the way that the gizzard forms, it kind of keeps some grit often in it, or just there's lots of little nooks and crannies, so sometimes the plastic can get stuck. So plastic's a huge ordeal when it comes to certain species of albatross breeding in the Pacific. So a lot of the chicks actually die because they get fed so much plastic that they don't know that they're still hungry, and they're basically getting fed non-food. But with our guys, that's probably not an issue, at least not at this age. But the problem is a lot of the plastics really chemicals and basically chemicals that can be poisonous but also interfere with their hormonal system. So overall, it's not good for them.
So that's incredible then. The plastics that they ingest might actually interfere with their ability to reproduce. Is that what you mean by hormonal system? There is starting some research coming out right now. None with prions that I know of just yet, but it is, it could basically impact their whole biology, really. And is this a growing problem for us in New Zealand or globally?
It's definitely a growing problem. Basically put away your plastic, people. Recycle. But it's also, that's one of the things we want to look at is whether there's time difference. So Harper did a great study, and he looked at storm wreck prions as well in New Zealand. And so we have a measure to compare from like the 50s to the 70s. And so it'll be interesting to see whether or not it's increased. So back then he was finding about 15% of the birds had some sort of plastic in their digestive system. And so if I was a betting woman, I'd probably say we have more now.
And so you're intending, once you've taken all these measurements, to publish that data, and it will be available for us to find out and do some comparative work on it?
Oh yeah, most definitely. I think that's kind of the really important thing is, unless you publish it, nobody really knows. It wasn't really done. So, I mean, if I know the story, that's not enough. But we'll also be putting a blog out there as well. So.
Thanks, Sarah. That'll be great. And I think that is a really timely reminder for us to think about how much plastic we use and think about as well where it ends up in our ecosystem and what other birds it could be affecting. Mm-hmm. Now, just before we hand back over to Scott, I've got one more question for you, if that's okay.
Sure.
So this is from Will Roberts at Plateau School, and thank you very much for sending in your questions. He says, is there a common age of birds found? And I'm presuming that means birds from the July 2011 wreck.
Yeah, what we were hoping for that we'd have a lot of immatures because, like we said before, they'd have a low survivorship anyhow. But what we were finding with the broadbill prions, the ones that was, most of the wreck, it's mostly adult females. Well, not mostly, but there's a large bias towards adult females, which has long-term implications on the population. Some of the other species, like the salvins that were examined, it's mostly immature males, so we're not as worried about those guys.
Okay, thank you. So in answer to your question, Will, it looks like, unfortunately, there are a lot of adult females in this particular storm-wrecked population. Mm-hmm. So thanks very much for your time, Sarah, and that was really interesting insight into your research.
Thank you.
So finally, we'll make one more cross the lab to Scott, who's going to be there with Susan Waugh talking about the collections.
That's right. Thank you, Ruth. And thank you, Sarah. So like Ruth said, with me again, you met her earlier on, is our Senior Curator of Science, Susan Waugh.
Hi,
Welcome back, Susan.
Hi, how are you?
Good, thank you. So before we ask you a few questions, we've got another question that's come in from Mrs. Brattle in Kiwi Room at Plateau School, and she's asked, and she'd like to know, what subjects do you have to do at school to become a scientist like you?
Oh, that's a great question. Well, I did, at my high school years, I did quite a lot of science subjects. I did chemistry and biology. But in addition to doing, studying things in detail, you also need to be able to explain your work to people. So having good English skills and speaking skills is really important. I sometimes wish that I'd done more drama or something to help me do public speaking a bit more than I do at the moment. But the other great thing about being a scientist is you actually get to be quite creative. And so it's good to have creative skills like music or art, because actually you have to use your brain and think up different ways to solve problems. And I think it's a great mix as a career of ways to use different parts of your personality and different skills that you have.
Brilliant, thanks, Sue. So not just science, make sure you're studying other things as well, like English and creative subjects like art or drama maybe. Great. So during our talk we've been talking about our collection and how a lot of these prions will end up in the collection here at Te Papa. So I'm going to ask you a few questions about our collection, Sue. So we've got a table wheel in here with a very, very small part of our collection, no doubt. First of all, our collection at Te Papa, our sciences collection here at Te Papa, why is it so important?
Well, we have about 500,000 specimen lots that are registered, so different parts of the collection are referenced under numbers to help us find them and store information about them. And they're really like an encyclopedia of the life that's here in New Zealand, so both plants, animals and invertebrates. And so it really helps us understand all the kinds of plants and animals we have and how they're different from each other, why some are found in some parts of New Zealand and others not, and so forth like that.
So our collection, we try to collect specimens of every living creature in New Zealand, every plant, every animal, is that correct?
Yeah, that's true. We try and have at least one of everything, and obviously it's a big task because in specifically the invertebrate groups, there's many hundreds of thousands or even millions of different creatures amongst the worms, the tiny shells, different little bugs that occur. So we really only have quite small proportions of the total New Zealand fauna of some of those groups, but actually for birds and mammals, we have virtually everything that has occurred in New Zealand. And in fact, for the bird collection here, we have the largest collection internationally of New Zealand birds. We have around 26,000 specimens of birds in our collection, and that's made up of specimens that are kept in different ways. So earlier we talked about the skins that we have, which are these ones. So the little bird is lying on its back and its feet and its wings are stretched out, and that means it's actually much easier for us to take measurements of critical things like how long their legs are and how wide the bill is. But for display and for helping us interpret how the species live in the wild, it's more useful for us to keep some specimens like this, which are called mounts. So they're taxidermied, just the same, they're preserved, dried, and the skin of the animal has been prepared so that it doesn't rot. But we keep a few of these to help people understand what they look like when they're alive and the ways they fit into the ecosystem. And then we also keep specimens that are just the bones, so the skeleton collection. So we have both animals that are alive today, and we also have many, many fossil birds from New Zealand. We have about 40,000 different specimens that are from animals that don't occur anymore, they're dead and they've become extinct. So for example, moas and the Harpagornis eagle, and some of the extinct birds that have recently been discovered and described by Te Papa's team, so things like adze, bills and the northern takahi. And then the final sort of collection we have are these spirit collections. So here we've got some stomachs of the birds, such as the ones that Lizzie and Sarah were talking about recently. But we also keep whole birds, and these are probably ones that maybe weren't in as good condition, they're maybe a bit rotten, but we can still learn a lot about the animal by keeping it pickled in a jar, basically.
So there's basically four different types of specimens, or four ways that we preserve things here at Te Papa.
Yeah, that's right, and we put those to different uses depending on the kinds of science we're doing. So for example, if you came across a deposit where there was soil and bones of creatures all mixed up together, chances are that the birds in that deposit would be in skeleton form, and it's much easier for us to see if these bones match, if they're already bones, than if we're trying to match it with a bird that's actually got all its feathers and skin on still. So yeah, we have different purposes for the different kinds of collection specimens we have.
Okay, so the ones that I'm sure most viewers would be familiar with would be things like the mounts here. They're the ones that we'd see in the museum that visitors can come and look at, whereas the other ones are primarily used for research. Would that be correct?
Yeah, that's right, and probably the majority of our collection are really designed for scientists to use to help understand, kind of unlock the secrets of the species. But it's also interesting for the broader public to understand how we use these specimens and what the value is in terms of understanding how the species relate to each other and what kinds of different plants and animals there are in New Zealand.
Brilliant. So I think you answered it earlier, but the number slips my mind. Can you just tell us again how many specimens we have in our science collection?
So we have about 500,000 when you consider the plants and the invertebrates and the vertebrate animals. We have about 26,000 bird specimens. And of the prions, we've actually got a really important collection at an international scale. We've got about 1,200 here. And so this new group of specimens coming at 800 is actually a really important addition to the group of birds we already have. So people come from all over the world to study our collections, and on particular areas, so for example the fish collection that we have and the birds, and particularly albatrosses and petrels, we have really top ranking collections at a world scale.
Really good. Thank you very much, Susan, for your time.
Great.
So I think we're coming close to the end of our time now, so welcome back, Ruth. We've spent an hour talking to various scientists involved in the project about these washed up prions, and we've learnt lots and lots of things. I've learnt lots of things. I'm sure Ruth has too. Can you tell us maybe, Ruth, what was the most interesting thing you've learnt today?
I think there was a variety of things, really. I was very impressed with Lizzie's quick dissection skills. I certainly don't think I could dissect 500 prions as quickly as she had. I'm also very impressed with Sarah's research. It was really interesting to find out a bit more about that, but particularly a really timely reminder for us to think about our plastic, our use of plastic, and how we should consider the ways in which we recycle it. But finally, when you were talking to Susan, I really liked her turn of phrase. She had a lovely turn of phrase saying we use these specimens to unlock the secrets of these species. And that's just a really great idea and a really lovely way in which the public can find out more about these species here at the museum. So I think that's it for our live stream here. But don't forget that you can keep asking your questions on Twitter using the hashtag ScienceLiveTopapa. You can email sciencelive@topapa.govt.nz, and also you can follow us on our blog, because science happens here every single day here in the museum. So you can follow our scientists for updates on the blog, which is www.blog.topapa.govt.nz.
And of course, you can come and visit Te Papa here in Wellington and have a look at some of our collection on display and see some of the science that's going on here and the science that we're explaining and telling everybody who comes to the museum. So thanks again for being part of our event today. We've enjoyed having your company. I hope you've learnt just as many new things as I have. And we might see you next time for our next live science event here at Te Papa. Thank you very much and goodbye.